All right, well, thank you. I promise not to bite if anybody wants to move closer to the front. So thanks for that warm welcome. Thanks for having me out. Having a great time. Saw some of you last night, I believe. So we're going to talk about cognitive psychopharmacology today. Maybe, if I can get my slide thing to work. Having all sorts of technical difficulties with my life today. Okay, come now. There we go. All right. So what we're going to talk about today, I'm going to do a brief sort of introduction as to why I think this is an interesting topic and why people should be interested in doing this kind of research. I'm going to start off talking about drug-induced amnesia, which is not as scary as it sounds. Talk a little bit about some of my work in nicotine withdrawal, and then talk a little bit about some attempts at cognitive enhancement. We'll spend most of our time in drug-induced amnesia and nicotine withdrawal because that's where most of my work is. But I do want to talk a little bit about some areas in cognitive enhancement and some new horizons to look forward to. Talk a little bit about some of the methodological challenges in this field, and then just kind of wrap things up. So let's start with a brief introduction. I want to first of all thank Elliot Hirschman down at San Diego State now. He's the one who taught me all about this field, introduced me to this field. So he's really done quite a bit to get this whole field started. In fact, he's got a great review article on cognitive psychopharm if you want to read that. So there are two main reasons to explore the links between drugs and cognition. The first of these is some applied questions we can answer, and the second is we can answer a lot of theoretical questions about neuroscience and particularly the effects of drugs on human behavior. So the kinds of applied questions we might ask are things like, what are the cognitive sequelae of drugs of use and abuse? So what happens when someone smokes marijuana? What happens when someone smokes nicotine or does cocaine or amphetamines? Tons of research in this area, so lots of important consequences of that. More recently, a lot of people have turned their attention to what happens when someone tries to get off of those drugs. So we're going to talk about what happens when you try to get off of nicotine. We'll talk briefly about some effects of people who are coming off of crystal meth abuse. Other kind of applied questions we can ask: can we improve cognition in healthy adults or older adults? Can we prevent cognitive decline in older adults? So these are obviously important general application questions about how we might apply questions of cognition. Importantly, we can also answer a lot of theoretical questions about some issues using this particular technique. So this provides things like an experimental model of amnesia. The problem with studying amnesia patients is, one, they're very hard to find. So you have to find somebody who has amnesia. You also don't have very much control over the situation. You just have an amnesia patient to work with to try to compare with non-amnesic patients. Whereas in drug-induced amnesia, you can actually experimentally manipulate amnesia in a way you can't do in other kinds of studies. So this allows systematic investigations of dissociations of things like conscious and unconscious components of cognition. One of the things we know about amnesia patients is they tend to have intact unconscious cognitive processes like implicit memory. Terrific work from Barbara Milner, working with patient HM, showing that HM, while he couldn't remember anything about his recent episodes, he could still learn new things. And so we know there are conscious and unconscious components. And this particular technique provides us with a way to investigate that phenomenon experimentally. So it also allows us, on that same note, to investigate specific hypotheses regarding the role of neurotransmitter systems in healthy adults and what their role might be in cognition. So by introducing endogenous drug or exogenous drug, we can actually determine whether or not those drugs affect a specific cognitive process and make inferences about the neurotransmitter systems involved. So there really is a lot we can learn from this kind of model about how cognition occurs. So I want to start off talking about drug-induced amnesia. There are really two main targets for causing amnesia in healthy humans. The first of these is using anticholinergics. So they target acetylcholine, in particular the muscarinic drug scopolamine. Marion up at Johns Hopkins does some really interesting work working with scopolamine. It's got some pharmacokinetic issues that make it a little harder to work with, but it is an interesting drug. Some of you may have actually taken it. It's a drug often given for seasickness. In fact, that's its primary use. But it does cause some pretty dense amnesia. Our target in the studies I'm going to talk about today is gamma-aminobutyric acid, or GABA. And all the drugs listed here are GABA agonists. So scopolamine is an anticholinergic drug, whereas all of these drugs facilitate the action of GABA. And GABA is, of course, an inhibitory neurotransmitter that shuts down or at least inhibits long-term potentiation, particularly in the hippocampus, which, of course, is the process by which we think long-term memories are formed. And so by looking at these different kinds of drugs, we can look at how GABA might be influencing memory in healthy adults. Each of these has different sort of what we call amnestic properties. So Xanax has some light amnesia, particularly in heavy doses. Most doses people take, they won't remember greatly, but they won't have dense amnesia. Ambien, or Zolpidem, is actually technically not a benzodiazepine, but it acts on the benzodiazepine receptor, so we just stick it in the same category. It works the same way. Its chemical structure is technically not a benzodiazepine, so I want to make that clear. But some interesting work, again, by Marion up at Hopkins on the cognitive effects of Zolpidem. You've probably heard news stories of people who wake up in the morning and don't remember going to the grocery store or cooking food or doing other sorts of things. And they talk about it as, somehow I moved off my slide. They talk about it as Ambien causing people to go out and sleepwalk and do all sorts of other things. That's not it. People do all these things while they're awake and conscious. They just don't remember having done so. And so it's really actually amnesia that's happening. Zolpidem also can cause hallucinations, which is unpleasant, having had that experience myself, which is why I don't take this particular drug when I try to get to sleep. Nothing worse than waking up thinking there's 5 people standing around your bed and they're not there. Triazolam is another one that's used a lot. Triazolam is used a lot in dental offices in conscious sedation procedures. But the drug we're going to talk about today is the sort of granddaddy of all amnesic-inducing drugs, called Midazolam. And Midazolam is used primarily in what are called conscious sedation procedures. And in these procedures, what's happening is you're usually awake for unpleasant procedures. So if you go in for something like having your wisdom teeth pulled or having an endoscopy where they're going to put a tube down your throat, you actually need to be awake for part of that. But in order to keep you from having anxiety and from having later trauma from the experience, they give you a pretty healthy dose of Midazolam. In fact, one of the great things about this particular drug is it's really safe to use. And so in the studies I'm going to talk about, we use a pretty low dose. If you were to go in for an outpatient surgery, they would just give you a vial of it, just because it's that safe. It's immediately reversible with a drug called flumazenil. So from a research perspective, this is a terrific drug. It's safe. It's used a lot. It's immediately reversible with another injection. So it works really well for us. So we model our research studies on these conscious sedation procedures. So all the studies I'm going to talk about occur in a post-anesthesia care unit, which we call the PACU. The studies that I was involved with were at GW. One of the things I want to talk briefly about are the pharmacokinetic properties of Midazolam, because it really makes it uniquely suited for this kind of drug-induced amnesia study. Most of the other benzodiazepines I've talked about are primarily given orally in a pill form, and that makes their properties really unstable. So it depends on what the person's eaten, their gender, there are all sorts of things as to how quickly they're going to absorb drug from their GI tract. But we use Midazolam, which has these great pharmacokinetic properties. So pharmacokinetics, when we talk about that, this is how drugs are handled by the body. So all drugs have some sort of route of administration. Midazolam is primarily intravenous. It is what we call water-soluble. And water-soluble drugs are usually introduced either intravenously or intranasally. And in fact, Midazolam is available in a nasal spray, which I find, I don't know about that. But it's a very good, one of the biggest things this is also used for is to stop seizures. And so it's a really good anti-seizure drug. And so they oftentimes will give people with seizure disorders a nasal form of Midazolam, but then I have this vision of somebody being on the train going somewhere, having a seizure, and then waking up and having no idea where they were because they just dosed themselves with Midazolam. So when we talk about pharmacokinetics, we're talking about these absorption properties and how quickly they are eliminated. So Midazolam is water-soluble, easily introduced intravenously. It's incredibly fast-acting. Within 5 minutes, it reaches what we call peak plasma concentration. So it's pretty much gone everywhere it's going to be. And the patient, or in this case participant, is slightly sedated and won't remember much after that for the next 15 or 20 minutes. So it's very rapid acting. And the other thing that happens is it's rapidly metabolized. All drugs, except for alcohol, have what we call a metabolic half-life. So it's basically how long it takes to eliminate half the dose of the drug. And midazolam has like a 60, 70-minute half-life. So it's really rapid. Benzodiazepines like Xanax, Ativan have 8 and 10-hour half-lives. Midazolam's in and out pretty quickly. So it allows us to set up these very rapid procedures. So this is basically how one of these studies works out. We've got a participant that arrives. We take their vital signs, make sure they're healthy. We've already asked them a zillion questions to make sure that they're healthy, they're not pregnant, they're not considering getting pregnant, all sorts of things. They get an IV catheter by the anesthesiologist. They get 3 mics per kilo, which is a pretty low dose, or 10 milligrams of saline. So these are all double-blind, placebo-controlled crossover designs. So all of our participants come in for one session and get midazolam, and one session they get saline. As the investigator, I handed the anesthesiologist an envelope. They went off, came back with a syringe, injected the drug. And theoretically, it's double-blind. Now, for about half our participants, I couldn't tell when they got the midazolam. For the other half, pretty clear. Because the drug goes in and they go, or they just fall asleep. So at that point, you have to just pay them and send them home after they've had a chance to nap. So then 5 minutes later, we're off and running. Study phase begins. About 15 to 18 minutes after injection, their study phase is over. Then we have this retention interval where we do a bunch of other stuff. And then 70 minutes after the injection, we start their test. So at this point, at 70 minutes, drug's worn off. Okay? So the drug is actually only acting for these 15 to 18 minutes. And so we get pretty dense amnesia for that time period. So that's the other great thing about this is very short acting. There's no long-term side effects. And so we can actually get in and out pretty quickly. So I want to run you through some of the studies in this area. These first several I'm going to talk about I wasn't involved with, but they're important to sort of set up why I think this is an important field. And so we'll talk our way through these pretty quickly. Elliot and his colleagues, before I started working with them, were really interested in using this as a way to explore conscious and unconscious components of memory. So explicit memory is, of course, conscious forms of memory. Implicit memory is unconscious forms of memory. And so they started off with just some basic implicit memory tasks. So they use what we call perceptual identification, which is you rapidly present a word, and then they see if the person can name it. And words that they have studied under midazolam, they're more likely, or words that they've studied earlier, they're more likely to identify, but they're not consciously aware of the fact that one was presented earlier. And then word fragment completion, they complete it with the first word that comes to mind. So these are tests that don't require any conscious effort, but the interest then is do people use explicit memory? That is, do they consciously remember part of the study list to accomplish them? And what you can see is, if you look at the midazolam versus saline, there is a pretty significant reduction for these seen items, for the previously studied items. So these are studied under saline, these are studied under midazolam. They're less likely to identify them. They're still identifying them, but at a lower rate. And it's the same thing with word fragment completion. The really incredible thing about this data to me is how consistent the pattern is across all the conditions. So you get this minor reduction, and that tells us that most implicit memory tasks have a conscious component. That is, people are using their conscious awareness to answer those questions. So when we talk about using an experiment to test implicit memory, we're probably really not doing that. And that's what I think the importance of this data shows. So then Jason Arndt, one of our colleagues from Middlebury, conducted this pretty great, I think this was his dissertation or somewhere in there, where they did category cued recall and category free recall. You can see free recall under midazolam, where we're trying to remember something from the study list, is abysmal. In fact, it's almost non-existent. These were category generation items, so where they were given a category cue, so a little bit better when they were given a category cue. But then we go to the implicit memory tasks, and they're actually much better. So again, shows that midazolam is affecting primarily conscious components of memory, but not the implicit or unconscious components of memory. finally, and these are the last two studies I want to talk about that I was involved with, they started looking at what's called the process dissociation procedure, the process dissociation framework. And I want to review this briefly because it's a pretty commonly used procedure that we're going to see may have some issues. So in a cube recall task, participants are instructed to specifically exclude studied items. So they're given, say, read and generate word pairs, and so they'll be given the cue like hot, and maybe at study they got cold, and they're told specifically to exclude whatever word they generated at study. So don't tell us that word, give us another word. Whereas in the inclusion condition, they're just told, give us the first word that comes to mind, whether it's one you studied or not. The idea here is that the exclusion condition is entirely conscious, whereas the inclusion condition includes both conscious and unconscious components. We can use some math to derive the conscious components. So we take the inclusion minus the exclusion, we end up with conscious component of the task. Okay, everybody follow that? I've simplified down this entire giant, hard-to-read Jacobi paper into 3 sentences. So hopefully that worked. Now, if Larry was here, he'd probably tell us that we got it all wrong. But so that's the basic idea. And what they found is under the exclusion condition, this is really interesting. Participants were much more likely to come up with an item from the study list, even though they were specifically told to exclude them. So that means that they don't remember that that was from the study list. Okay? Under the inclusion conditions, they're actually producing fewer items because under the saline conditions, they can consciously remember the ones they studied, and so they're much more likely to come up with those. So again, pretty interesting procedure showing conscious and unconscious components. Well then, Marion and Elliott and one other person did another study using this sort of updated process dissociation procedure to look at recollective and automatic processes. And what they discovered is using those calculations, Midazolam actually facilitated automatic processes, which is really unlikely. That is, it's very unlikely that a drug that causes amnesia is going to make your memory better. And so what they were able to demonstrate with this particular experiment is there's a flaw in this framework in terms of all of its mathematical assumptions. The reason I wanted to talk about this work is it demonstrates the really important theoretical questions you can answer using this kind of procedure. And so it really is a unique way of doing that. Okay. Any questions about any of that? All right, well, let's get into the stuff I know more about, which is the experiments I conducted. So the first one of these is called Metamemory without the memory. So an important clinical consideration in these conscious sedation procedures is when a patient wakes up and you're giving them instructions and they're answering things, do they have any idea that they're not going to remember it later? Right? It's an important question because staff doesn't know that there's anything wrong. Family doesn't know there's anything wrong. They're answering questions. And I can tell you patients under Midazolam can answer questions on the computer. They can talk to you. They can do all kinds of things and have no idea that it happened. It's really kind of freaky. So often patients show no outward signs of sedation. So then, of course, of theoretical importance, while we're on the same topic, is whether or not things like judgments of learning are based on some sort of direct assessment of memory formation. So one of the theories of metacognition is that people directly access their memory to try to decide whether or not their memory is correct. So here's what we did. Here's the basic study design. Given Q target pairs, and they make a judgment of learning. They just say, how likely are you to remember this later? Then we move on to the test phase. They get the cube recall, so they get the Q, try to recall a word. Then they tell us how confident they were in that. We're actually not going to look at that data because it turned out not to be very interesting. Actually, we'll look at it briefly. If the item they recalled was correct, they went on to the next trial. If the item they got was incorrect, they then did what's called a feeling of knowing judgment, which is they tell us how much they feel that they might be able to find the answer when we give them 4 alternatives. So they've got 4 choices. They tell us, I feel like I'll know this. And then they pick one of those, and then they move on to the next one. So we do judgments of learning, retrospective confidence, and these feeling of knowing judgments. So we have kind of a lot of data to talk about fairly quickly. So here's the cube recall data. You can see under Midazolam, participants are recalling, I think it's like 0.03% of items. So basically 0. It's not really significantly different from 0. Whereas under saline, they're getting about a quarter of the items right. People are pretty bad at cube recall. It's a pretty tough task. So then when they're given that 4-choice recognition task. They're a little bit above chance. In fact, they are significantly above chance for midazolam. So they're able to kind of find their way through to the right answer some of the time. Whereas, of course, under saline, they're almost 60% correct. So it's still impacting their memory, but they've got something in there. They can still access some of that memory. It's just pretty significantly degraded. So we have cube recall essentially eliminated and recognition memory reduced to above chance levels. So then we look at their judgments of learning. Under midazolam, they're ironically higher than when they are giving them under saline. Let me explain the reason for that. So remember I told you that each participant's tested twice. So they come in once and are tested under saline, and they come in once and are tested under midazolam. Well, we counterbalance order. So half the participants come in and get midazolam first, and then they come and get saline second. Well, this effect, this lower judgments of learning in the saline condition, is entirely accounted for by the people who got midazolam in the first session. Then they come back in a week later and say, I sucked at this task. I couldn't remember anything. And so they don't realize that it's because they were under the drug's influence. They just think they suck at the task, and so they reduce their judgments of learning. Really interesting finding. So then you can see their retrospective confidence. That is, after they have discovered they have no memory, pretty low compared to under saline. And again, same with their feeling of knowing, pretty much reduced. If we look at what are called calibration curves, so a perfect calibration curve would be along this diagonal line. Even under saline, they're wildly overconfident. Whereas under midazolam, they do know which ones, the ones that they're highly confident in. So these are the ones they rated 80 to 100% that they were going to get correct. They're much more likely to get those right. So they're getting about 15% of those right, which under midazolam is kind of stellar performance. It's about 15 times the rest of these. So they actually do pretty well on those ones that were highly confident in. So there may have been something very distinctive about those pairs, something like that. So the conclusion here is that participants who are under the influence of a drug like midazolam, or have just taken a Xanax or an Ativan at home or an Ambien, have no idea that there's anything wrong with their memory. And in fact, this particular procedure, it turned out 1 year at a conference, 3 of us did this exact same thing with 3 different benzodiazepines, and we all got the same results. And at Psychonomics, so there was the 3 of us all in a row. And so it was actually a really fun conference. We actually sit around and talk about our drugs in metamemory. So again, turns out to be an important component. Also turns out that tells us a lot about how metamemory works. It's not that we're directly accessing our memory. It's that we're judging whether or not we think we might remember something later based on the perceptual properties of that stimulus. The next study I want to talk about is looking at episodic contributions to semantic memory. So one of the things we know is that a lot of the things you learn start out being episodic memory. That is, you remember the time and the place that you learned them. So tomorrow, you'll be able to tell your friends, oh, I went to this talk from this guy talking about drug-induced amnesia. You'll remember being here. Now, you probably all know the first president of the United States was George Washington. None of you, I would imagine, can tell me when and where you were when you learned that fact. That fact has become independent of its context. So we're interested in that process. That's how do things go from being an episode to just something you know. So here's what we did. Came up with this rather intricate procedure. It might take me a minute. So I came up with a bunch of semantic facts that no one would know. So one of them was the capital of Botswana is Gaborone. My other favorite was Donald Duck's middle name is Fauntleroy, which is true. So they get this fact. Then right after that, they get what is the capital of Botswana. And if they get it right, then I would code yes or no. They did each of these 50 facts twice through that procedure. So it's a pretty robust encoding condition, right? Half of the participants got this semantic retrieval condition. Answer any questions you might know the answer to. The other half of our participants did this episodic plus semantic retrieval condition, but they should only answer questions they know were presented earlier. Essentially, this is the inclusion-exclusion we talked about earlier, but with a semantic memory task. Okay? So that's what we did. And what we found, interestingly, is when we asked participants to reinstate their context, that is, use their episodic memory, they were much more likely to remember those items studied under midazolam. Now, it had no effect under the saline conditions, that is, when their memory was intact, didn't help them at all. But when their memory was degraded, they did much better. So what that tells us is, under those degraded memory conditions, that episodic context is a really important memory cue. So what I've started telling my students is, if you're in an exam and you know you studied something but you can't quite remember it, try to reinstate the context you were in when you were studying it. Because what you're trying to do is reinstate that, where you were, that episodic context, and use those cues. Now, whether it works for them or not, I'm not sure, but it seems like a good idea. So this is consistent with some results from other people. And essentially what we're finding out is that by trying to use the source of a memory as an episodic memory cue, you can actually identify that memory and do much better. Okay. Last thing I want to note is Lynn Reeder and a bunch of her colleagues at Carnegie Mellon have actually done some fMRI work in this area, and they found that the area that primarily gets sort of shut down during midazolam study sessions is the left dorsolateral prefrontal cortex in this particular area right here, and that seems to be part of this area that's responsible for binding things to their contexts. In fact, that's what their experimental results show as well. And so it appears that one of the things that this drug disrupts is this encoding of context and binding things, novel associations together. And again, that's consistent with all of the data we've seen so far. Okay. Questions about that? Okay, well, this is a really exciting and interesting area. I'd love to get back into doing this research now that I'm back at a medical center, because there's just so many cool things you can do with it. So I encourage, if you're interested in this, there are certainly people working in this area for you to get in touch with. So I want to change gears just a little bit and talk instead about administering a drug, taking somebody who's already using a drug and taking it away from them. Essentially, this is a much easier thing to do because it's a lot easier to find people who are smoking and get them to not smoke than it is to get them into a hospital and inject them with drugs, right? Because they're already smoking. Although finding smokers has gotten harder and harder, which is a good thing. Now, from a research perspective, it's really tough to do this research because nobody smokes anymore. One of the things that we know with smokers is that there is some cognitive disruption that goes along with quitting smoking. That is, while people are trying to quit smoking, they have difficulty concentrating, they report being fuzzy-headed, that sort of thing. The great thing about this kind of research is, again, we have applied and theoretical questions. The applied question is, of course, if we can get people through this fuzzy-headed period, maybe we'll be better at getting them to stop smoking. The theoretical questions are, of course, what effect is nicotine having on memory? What are the neurotransmitter systems involved? How is that all occurring? And so there's important things to think about. So these cognitive impairments may contribute to difficulty in cessation efforts, and nicotinic receptors, which are the nicotinic acetylcholine receptors, might play an important role in cognition, and particularly if we start looking at the up and down regulation of these in chronic smokers. So these are some important questions to think about. So specifically, we're going to talk today about episodic memory. You might have noticed that seems to be where I spend most of my time. One of the things we know is that nicotine causes structural neurochemical changes in the hippocampus and related areas. In particular, what happens is the activation of nicotinic acetylcholine receptors, or NACHRs, actually downregulates the release of GABA. That is, it reduces the release of GABA in the hippocampus. So what's happening is people who are intaking nicotine, it's reducing their GABA levels, and as a result, some people actually get a little bit of a boost in memory under nicotine. The problem is, to give somebody who is nicotine naive enough to help their memory, you tend to make them very nauseous and very unhappy. So that's one of the things that's going on. So withdrawing nicotine, because now we've taken away that downregulation of GABA, it actually causes an upregulation compared to baseline. So we now have an increase in GABA compared to baseline. And as a result, we would expect reductions in memory or memory impairment, as we've just seen with all the midazolam work, because that's what midazolam is doing, is affecting GABA in particular. So that's why we expected there to be some episodic memory impairment from this kind of work. So purpose of this work I'm going to talk about specifically today, one of the things that happens in nicotine withdrawal is some disruptions in attention, particularly sustained attention. And of course, there are different components of attention. Sustained attention is one, selective attention is another. And so the question is whether or not memory impairments have any attentional component. Because if you're not selecting something from the environment to pay attention to it, then obviously you're not going to remember it later. So in this previous study I'll talk about here in a minute, we did see some reductions in this kind of sustained attention task. So our goal in this first study was to determine if reductions in memory have an attentional component to them. So basically what we did is we included an attentional manipulation along with our drug manipulation to make things very complicated to see whether or not there might be some sort of interaction effect. So what we have here are moderate to heavy smokers that come in again for 2 sessions. We're very big on within subjects designs in my world because it's much easier to get people to come in twice than to find twice as many people. So they come in and smoke what we call ad libitum. They smoke as much as they want coming up until the time they come in, right before they come in, doesn't matter. And then we have abstinent. They're asked to refrain from smoking for 24 hours. You have to pay people to do this. And in fact, we lose about a quarter of our participants because they can't make it. We lose a few more participants because they quit smoking for 24 hours and then they just never come back because they never start smoking again, which is great. So I don't mind that. I mean, it sucks because then we never get them in their smoking condition, but you know, we're happy for them. They're going to live longer, and so that we get some karma points for that or something. So this allows participants to act as their own control. So we're not trying to match people based on how much they smoke or don't smoke or their age or any of that stuff. So it allows us to do some more stuff. So we verify their abstinence, their tobacco abstinence with a breathalyzer. You can actually measure carbon monoxide in everyone's breath, but it's pretty high in smokers. There are other ways to do this, but this is the least intrusive. The other is continine measures, which either is urinary or salivary, and I'd rather do the breathalyzer. It involves much less work. So we include a variety of tasks along with our episodic memory task to try to see if there are other secondary effects of withdrawing from nicotine. So you've got them in the lab, you might as well test them a bunch. So we do short-term working memory, attention, that kind of stuff. So the first study I talked about, we saw reductions in recognition memory performance and increased reaction times in a vigilance task, which is where we ended up with in the study I'm going to talk about now. So we also got no reductions in short-term memory using a basic digit span task. We included that again just to make sure that that was correct. So overall, general global results from our study, really significant reduction in memory performance. These are actually D primes. So we get pretty major, it's about a 20% reduction in memory performance, no effects on short-term memory, no effects on working memory. So it's all happening here in episodic memory. Now to test whether or not there's an attentional component to this memory effect, we did a full versus divided attention manipulation. So using a procedure that Neil Mulligan came up with, words appear in the center of the screen that the participants are encoding, and then they have to do the secondary digit tracking task where there's digits around the word, and every time 3 odd digits come in a row, they have to push a button. So it's a pretty tough task. So what we see is when people are smoking as usual, pretty significant reduction in full versus divided attention. But there's no interaction here. That is, the reduction under tobacco abstinence is the same for full attention versus divided attention. So there's really no, we would expect if this was mediated by attention, you would expect a huge reduction in divided memory performance when people are abstinent from smoking. Now what's interesting about this is, let's skip over that slide, it's not that important, is we actually found a gender difference. And there's actually several people, this has actually become one of my most cited papers, because this has generated some really interesting questions. So for our males, pretty big attention difference when they're smoking as they normally are. I mean, that's a pretty big full versus divided attention. When they're abstinent from tobacco, their divided attention performance doesn't change at all, but their full attention just plummets down to the basic same level as divided attention. Whereas in our female participants, we get a slight reduction in divided attention under abstinent conditions, but no change under full attention. So there seems to be this interaction between sex and attention. I would like to tell you that I know why this is happening, but I don't, just that it is, and it's a pretty interesting finding. To give you some idea about how controlled we actually were able to do this analysis because we happened to have equal numbers of males and females. We didn't on purpose do this, it just happened that it came out that way. And we also keep really meticulous records. So one of the things we had to do was determine how many of our participants were on birth control, because that has important effects on cognition, and also whether or not we had women in different parts of their menstrual cycle in the abstinent and ad libitum conditions, because that also has some important components. And so fortunately I had asked about all of those things. And went back through and added all that up, and it turned out that it all came out fine. We didn't have any differences, but it does provide this really interesting question about why there are sex differences. Males smoke at almost twice the rate as females do, and interestingly, though, females tend to report more sort of cue-associated smoking. So they're out with their friends and drinking, and so they'll have a cigarette then, and they seem to have more of that. And so it may be part of that may be mnemonic. We don't know, but there are some important questions there. So this provides some really interesting theoretical questions. Is this modulated by sex differences in the brain, or is this just modulated by some other difference that we don't know about? It's hard to say, but it does provide probably more questions than answers. So our results here are consistent with the idea that memory impairment is due to reductions in attention, and it provides that nicotine influences memory directly. And again, it's probably via the nicotinic acetylcholine receptors and their effects on GABA. So there is clear evidence that these particular neurotransmitter systems have important roles to play in memory. And we've known, of course, that acetylcholine is an important part of memory, but trying to tease all that apart and which components are involved is a particular part that we're still interested in looking at. So these are our working memory tasks. No difference between abstinent and ad libitum smoking. And our general conclusions are tobacco abstinence causes reductions in episodic memory performance, and again has important implications for cessation efforts. And drugs targeting those nicotinic receptors may be a viable solution for treating this kind of memory loss. And in fact, Chantix, which is varenicline, does seem to improve some memory performance during this abstinent period. And so people who can take something like Chantix will actually benefit from it. Now, one of the big problems with that is not everyone responds to those drugs, and we're going to talk here in a minute about pharmacokinetics and pharmacogenomics. And part of that has to do with there is actually a genotype that has a different susceptibility to nicotine, and those people do not respond at all to Chantix because for some reason it has something to do, my understanding is, I'm still trying to learn pharmacogenetics, and it's a tough road at my age, but you can learn anything at any age, but trying to learn the genetic stuff, there is something about the actual receptor surface in this genotype that's different, and so nicotine binds differently with that receptor site, is my understanding. Now, I might not have gotten that out right, but it is an interesting component, and it's something worth talking about. So in the time remaining, I want to talk a little bit about some questions of whether or not we can actually enhance cognition. And this is, of course, an important question. So obviously, if we're going to have try to help people who are aging improve their cognition, that might be something we can do. So there have been attempts to use things like nicotine, but in nicotine-naive subjects, you end up with dizzy, cranky people, and so it doesn't work well. We've tried using a hormonal supplement called DHEA, which is dehydroepiandrosterone, to try to improve cognition in postmenopausal women. To, as you'll see here in a minute, very little success. To give you a quick overview of DHEA, it's an abundant steroid hormone. I'm going to skip through this quickly because this is the less interesting part. DHEA is available over the counter. You can get it at GNC. The problem is you can take this and it will become all this other stuff. So if, for example, you want to take DHEA because you want to build more muscle, have more testosterone, that might happen. Or you might end up with more estrone, estradiol, and estriol. Not going to help you build muscle. If you're trying to improve memory, you want more estrogen because that's going to improve your memory and benefit your hippocampus. But if you turn it all into testosterone and androstenedione, it doesn't do you any good. So what we've done, and this declines over time, what we did is we actually measured circulating hormones. This is one of the first studies I did with Elliot. We actually brought participants in. This was a huge study. They came in twice over an entire day. And so they were tested at 9, 10, noon, and at 4 p.m. After taking DHEA for 4 weeks or taking a placebo for 4 weeks. And they were tested each of these times on about 8 different tasks. So I programmed a lot of tasks in this particular study. And we also measured their circulating blood levels. So you can see DHEA increased their estriol, their free testosterone, their DHEA, their DHEAS, total testosterone, and estrone. All these hormone levels increased in these postmenopausal women. We use this particular participant pool because they're sort of like a hormonal blank slate. Because they're basically very low on all these hormone levels. So you're going to get effects across the board. So you can see they're very low on all of these, and we got some pretty nice effects. We ended up combining estrogens and androgens together to try to see how they were related to cognitive performance. We found that estrogens were positively associated with memory. Androgens were negatively associated with memory. Estrogens were negatively associated with perceptual identification, and testosterones were negatively associated with attentional vigilance. And so there are some interesting correlations between these circulating hormones, but at the end of the day, none of these were improved by adding DHEA to the diet of these participants. So it's not something to take for improvement. So boost levels of estrogen and testosterone, didn't do anything to cognition. Same thing we found in another study, and this was a huge study, like 16-week, 100 and some participants, no improvement at all. So DHEA is not something to take to improve your cognitive functioning. So if you have older adults that are taking it for that, there's probably better things to do with their money. So other targets for cognitive enhancement that I've not been involved with but I think are really exciting, we certainly know that there's noradrenergic responses, so norepinephrine is associated with memory. That is, when you have a high level of norepinephrine, you have pretty solid memories of those events. That's why high stress events are memorable. We know those memories are consolidated better. And so there's some questions about how maybe we can use that particular phenomenon to improve memory. So possible improvements in memory consolidation from D-amphetamine. There is some positive effects of learning from Ritalin, which is methylphenidate. We know there's a steady trade in these things on college campuses as study aids, but these all have really potential abuse problems, and they have pretty significant health problems as well. Both of these, methylphenidate and amphetamine, do things like activate your sympathetic nervous system, which takes blood away from your internal organs. It does all sorts of things, not particularly healthy. So in terms of trying to come up with a viable cognitive solution for improving cognition, this is not one of them. There is one on the horizon, though, called modafinil. And modafinil is actually is a drug to treat narcolepsy. But it turns out that it's got some pretty remarkable components. It directly inhibits central dopamine and noradrenaline uptake transporters. As a result, it results in elevation of serotonin, glutamate, histamine, and reduces concentrations of GABA. We're back at GABA again. It's one of those really important things. So we reduce GABA, probably going to improve memory. And in fact, modafinil intake appears to enhance executive functioning, including fluid intelligence, improve learning and memory, enhance attention in more complex tasks, with little side effects. There are no emotional changes. There is very little sleep disruption. It's pretty remarkable. Now, the problem is the question of how to ethically handle this question. And so there's some pretty interesting ethical panels about, is this something that should be available for something other than just narcolepsy? That is, should we make it available to everyone? Well, but is it only available to some people? It's a really interesting question, and I think that we're going to start to see this question as we start getting more and more compounds like this on the market. We're going to have to really come up with some way to regulate this marketplace, because obviously we could end up with a pretty solid black market trade in these kinds of drugs, which I guess is better than a black market trade in painkillers. I'm not going to deny that, but it's something to think about. Now, modafinil has had some other interesting uses. There's evidence that people who are withdrawing from cocaine and amphetamine use benefit from modafinil to try to improve their executive functioning. It might actually be useful for treating head injury patients and boosting their executive functioning. So these are all potential questions. This is a really great very systematic review of the entire literature of this drug, and it's huge. I was stunned. This is a new drug, and there is hundreds of studies out on this. So take a look at that. I think it's in European Neuropsychopharmacology or something. So that's modafinil. So there certainly is some hope or belief that we might have some cognitive enhancing drugs on the horizon, but we do have some really exciting new horizons. The most exciting and huge areas of research, so if you want to go into graduate work and make a ton of money working for a pharmaceutical company, pharmacogenetics and pharmacogenomics is a huge area. And most of this work is being done in targeted cancer treatments. So they run the entire genotype of a patient, and they find the best cancer treatment for that genotype. But now it turns out that there's a lot more to this. So we might be able to do things like genotype the best antidepressant, because that seems to be very clear that there is a clear antidepressant. We also know there's a clear genetic component to smoking, and so if we can find that genotype and find targeted drugs for that. So this is a really exciting area that I think has huge potential. The other thing that I think it points out is that we have to acknowledge there are huge individual differences in this area because genetics are such a big part of that. So other areas, pharmacological agents to alter memories. This is some wild stuff. There is in development techniques to essentially decouple fear responses from phobic stimuli. So if you think about a phobia response as a stimulus-response relationship, right, that's a learned response. Using this drug propranolol, which is anti-neurogenergic, they initiate that fear response, give them the drug, and then they come back and the fear response is gone, that it has been decoupled from the stimulus. It's astounding to me and kind of frightening, truth be told, because you have to wonder what else. What does that change about the person's personality? I don't know. We can get into an ethics discussion another time. And then there's this other thing I was talking about last night. In animal models, they've demonstrated this ability to turn memories on and off with a laser. Did you? So, yeah. So if you want to watch this, go online to YouTube. There's this great memory special called Memory Hackers. And they actually show, both of these things actually are in that Memory Hackers episode, and it's amazing. And this mouse, they turn the thing on, it's happy and go lucky. They turn it off, it's afraid. They turn it on, it's happy again. It's kind of freaky. And the guy that learns to no longer be afraid of spiders, it's actually pretty fun. It's a really interesting thing to watch. So I think the other area to look for in New Horizons is drugs targeting neurogenesis. Because there's new evidence that antidepressants can promote neurogenesis, and that depression, one of the biggest problems with depression is a reduction in astrocytes in the hippocampus. That paper just came out this week. So I think that's going to be an important area. And so I would say that's some place to look as well. I want to finish then with some quick notes if you want to do this kind of work. Know your pharmacokinetics. I reviewed a paper once where they were talking about they conducted their experiment after the drug had worn off, and I looked up the drug, and it turned out they conducted their entire experiment while the drug was at its most effective peak, because they just hadn't done any of their homework. So peak plasma concentration and plasma elimination half-life are very important. And the sad thing is I ended up reviewing that paper at 2 different journals and rejecting it both times. So that's a really important thing to know. Individual differences are really important. For example, there are sex differences in what's called alcohol dehydrogenase. So this is a enzyme that's in your digestive system. Men tend to have about twice as much. And so before you've even absorbed any alcohol, most men have eliminated about twice as much as women will because of alcohol dehydrogenase levels. People with red hair, this is fascinating, people with red hair require twice as much anesthesia because they're much more efficient, genuinely red hair, by the way, not dyed red hair. So people with genuinely red hair, there's something about that genotype that they are much more efficient metabolizers, and so they actually can metabolize drugs at a much faster rate. People who like grapefruit. Grapefruit, I know you guys think I'm crazy. Grapefruit is a cytochrome P3A inhibitor. And cytochrome P3A is one of the primary enzymes by which drugs are broken down by the liver. And so if you take Xanax and eat grapefruit, you're going to get about twice the wallop out of it. It's going to take twice as long for it to metabolize out because of the presence of that grapefruit juice. So a lot of drugs you're not supposed to take at all if you're eating grapefruit. So you have to ask participants a lot of questions. You have to ask them a lot. People who hate cilantro. I hate cilantro myself. Awful. I don't know why people put that vile weed and stuff. But I have that genotype. So that's one of the ways that you can check for genotype. And so it's possible that they might have some other genotype. So carefully constructed research designs are critical. And of course, safety is paramount. And so like the informed consent form for our last Midazolam study was 14 pages long. Yeah. Because there's a lot of stuff. By the way, if any of you go in for surgery and are given Midazolam, they're not going to tell you that it is associated with birth defects if you're male or female. So if you're somebody who's trying to have kids, Midazolam is not something you should have because they'll forget to tell you that. And it lasts for about a year. So keep that in mind. So I hope you guys enjoyed this kind of quick run through. We barely scratched the surface of this area. I teach an entire class on it. Here's all the ways you can get in touch with me. If you look for me on YouTube, I have my entire drugs and human behavior class is online. I'm in the process of updating that. So you can listen to me do all this. If this recorded properly, and I'm hoping it did, I'll post this as well. Anybody have any questions, stuff they want to know about? Yeah. So here's how it works. So they're actually genetic knockout mice. So they actually have added a bioluminescent algae gene to the mouse, and then they give it a drug, and then while it's under the influence of that drug, it actually codes the neurons with the bioluminescent gene. And so here's the thing. So then when they shine the laser in, it activates only those that have that color added to the neuron, and so it actually activates those neurons. It's crazy. Watch that part of it. I mean, it's like I just sat there and went, what? Because it's really pretty amazing. And so what that's going to mean for people, who knows? I mean, obviously we can't implant algae into people's genes, but yeah. Sorry, let's have a hard time. I could hear. So, what was your question?
Saying that this experiment is saying that they can activate the cortical cell
Yeah.
without activating the retrieval pathway.
Yeah.
And like no one knows how to do that.
No.
Usually the retrograde amnesia is caused by the
Yeah.
controlling the retrieval pathway.
Yeah. So nobody knows quite how to do that. And that's why retrograde amnesia is so rare, is because most people have anterograde amnesia because it's all about.
Then I don't know why the drugs can do that.
Yeah. Well, our drugs again only cause anterograde amnesia. Yeah. So this is all anterograde. So they give them the drug and then it blocks the encoding of those memories.
Oh, so
It's not anterograde. It's all retrograde. It's all while they're under the drug's always given during the study episode. Yeah.
Oh, that's scary.
Yeah. Now they say that scopolamine has a slight retrograde component, but it's very, very slight.
So the test used to assess the memory is not object naming. It's more like iron learning.
Right. So while they're under the drug, that's actually when they're encoding. So they see words on the screen. They're under the influence of the drug. Then it wears off. Then we test them.
Oh, okay.
And so it's actually the actual creation of the memory that's blocked.
Yeah. I guess I misheard it.
Yeah. That's what it's happening. Yeah. That's a good question. You bet. Yeah.
So I feel like it's inversely correlated GABA levels with stress because you mentioned how when people withdraw them, they contain their GABA levels.
Well, one of the things, yes, in fact.
It seemed a little counterintuitive.
Well, so GABA is an inhibitory neurotransmitter.
Right.
So when there's more GABA, there's less